And now it's my privilege. Now it's my privilege to welcome Mr. Kumar Balasubramanian, Senior Principal Biostatistician from the McMaster University. And special thanks for having made it in person for our workshop. Thank you so much, sir. And I'll hand over to sir for the next session.
You can call me Kumar. It's okay. You don't need to. Hello, all. Thank you. Thank you for the kind invite, and thank you, Vignesh, for organizing the whole event. And it is a privilege and honor to be here at the CMC Vellore. In the last 13 years, I've heard a lot about this, and 7 years from Shrikant, and last 3 years from two of your students, Stain Marie and Grace. So it's definitely a pleasure. And seeing at all the study proposals and everything, I can see it's a pretty elite group. And I think clinical trial in general is in safe hands from all the researchers who are sitting in front of us. So that way we are happy about this one. And I understand it's an amazing lunch organized by Vignesh, and it's nice time to sleep. So Shrikant gave me a right timing to talk about study conduct and about teach. Yes. So I won't take it wrong if someone wants to doze off or sleep. It's okay. It's still fine. I'll try my best not. I don't sleep. So so we are going I have a lot of slides on around 50 slides, and some of them I will try to skip if it's not relevant here. But primarily, I'm going to talk about the study conduct in clinical trial, ensuring integrity and safety. So the most important thing, as Ananta was mentioning when we started this, let's take a step back to that session to Ananta, because that's a continuation of this. So I'm just continuing what she has completed. So the most important is the safety of the patients. So we are highly privileged to have a patient level data. No one wants to go to a clinical trial. Tomorrow, someone stands outside and tell you, okay, I'm running a clinical trial. Can you please give your blood sample? Can you please take this drug? None of us in this room, I'm telling you right now, will not participate. So it is truly a privilege that patients are participating in a clinical trial for the betterment for them, mainly for the society and around the world too. It changes the practice. As a physician, each one of you know, you cannot give a drug to anyone without a proof that the drug is working properly, without any side effects. Of course, side effects comes with it. Safety comes with a problem with this. But are we making sure that we are doing a right job in running a clinical trial? And there is a lot of articles that is published during the COVID times where there was a need of running a trial, and people were just running trial without thinking through, just running on different trials on patients. Patients were also not thinking through. They were just enrolling themselves because they want the treatment for COVID. So many papers were published in Lancet and NEJM. The big journals didn't think through what papers they are publishing in, and that need to be retracted. And it never happened in NEJM. There are 8 papers have been retracted during the COVID period that has been published. So that's the kind of trials that we are looking at and making decisions on. So as a responsible researcher, it's very important to have a proper study conduct and making sure that you are following everything that is required. So we have divided the presentation into two items. One is the internal monitoring and then external monitoring. And the internal monitoring is a time where Ananta said we are going to recruit the patient, randomization list. And the first patient is randomized. From that point, we start the internal monitoring on it. And then the second topic would be about the external monitoring, which is primarily focused on DSMB. I am specialized on DSMBs, where I've sat on more than around 40 to 50 DSMB as a lead principal biostatistician. So I had a privilege of looking at all unblinded data on different studies. And so there is a whole lot of scope, and it is very important that as a researchers, as a statistician, probably there are 2 or 3 statisticians here. How many statisticians are here? 1, 2, 3, 4. 2, 3, 4. Yeah. Okay. So as a statistician, it's a highly a privilege to sit in those closed sessions, and you will be able to see the lot of data that will make you, and the perspective of looking at the data itself will change. So the internal monitoring depends highly on the PI and the study team. And I also see a lot of people are doctors here and the study team members. So their focus, their involvement in the internal monitoring will be really high. So we'll be touching base on recruitment, ensuring data quality, compliance with protocol, adherence to intervention, and adverse events and safety monitoring. So recruitment is crucial on a clinical trial and ensures a selection of eligible and diverse participants for effective results. So just going to the outside and say, okay, I'm going to run this trial and need these many patients, it's not that easy. It needs effort. It needs some strategies to even collect the patients, that is right patients to your study. There is a lot of fraudulent data out there in the market. When you involve a site which is not the right site, it can just spoil your study. Your dream of having a good clinical trial can be spoiled by just one particular site who have not even randomized a patient, but they're just having a fake data in it. So monitoring your data is extremely important. And how do you monitor is through the recruitment. It starts with the recruitment aspect of it. I'm going to show you a practical, a live recruitment report that is we are using in a study that is currently recruiting. And it might look detail oriented, and I will explain why we really need that one. And the most important aspect of that is the recruitment is the ethical consideration, and strategies should always prioritize ethical implication. You cannot just recruit people because I really want somebody from Vellore. You need to look at the multi-center. If you're running a multi-center trial, you need to be carefully considering where I'm going to recruit from. Is it the right population of people for the study? Because once the study is published, where they are going to use this drug or the device or the treatment, it cannot be just in Vellore. It's around the world. It's around the places. So you need to be very careful about where you're recruiting people and how you're recruiting people too. And the strategies, and Ananta has mentioned this earlier, it's tailor messages and specific demographic to effectively attract eligible participants. It happens, it need to be well thought through, and it need to start from the time you design the trial. From the day, yesterday evening, probably you all have sat and designed an hypothesis. A lot of times when you're designing an hypothesis, there is a whole lot of factors that you need to look into before even writing out the hypothesis over there. Or I'm going to test the superiority of this trial. Is it even feasible? Is it possible to recruit the patients? Is it possible to monitor the patients? What kind of questions I'm answering? I'm talking about smoking as a primary hypothesis. What is a population? Do they even give you the smoking information? We don't know that. So we need to be very careful about writing an hypothesis, not to write an hypothesis and then think about, okay, I will just recruit somebody. So you need to think through the whole process of this internal and external monitoring. So here I'm going to give you an example of the current trial that we are working on. I don't want to mention the name of the trial, but the misinformation in the recruitment, lack of interest, transparent communication is extremely important when it comes to recruitment. You should not be lying anything to the patients and say things that will attract them to participate in a trial. It's a highly sensitive information. We are running a trial on obesity. It's a cosmetic surgery, I would say. Technically, it's a cosmetic surgery to avoid cardiovascular death or cardiovascular disease, MACE outcome as an outcome. Now, how do you project this to a patient? You say, oh, you're obese. Oh, we have a surgery. It's cosmetic. It reduces his liposuction. You just remove the fat and everything. Or you say, we're running a trial that's going to save you from dying, like the cardiovascular disease. You may not get a MACE outcome. It's very sensitive thing that how do you want to project it? Are you going to hide or are you going to say which one first and which one second? So those information, eventually they will come to know. And that's a lot of dropout rate. And as you were mentioning, the dropout rate increases because when in the process of it, they will realize you didn't give me the whole information. You just told me that this is an info to avoid something. No, because a lot of times we are desperate when it comes to recruitment because we need patients. So we would tend to say anything and everything. It's possible to recruit people. So we need to be very honest. And the lack of interest just comes in as and when you need, as a research coordinators, they play a very major role. A lot of time they don't realize the PI is on one side, statistician on the other side. The most important role I feel is the research coordinators who are handling the study need to be 100% sure about what they are doing, and they need to be ethically understanding the concept, what can impact a trial. And addressing the concerns and everything is part of the process, and the transparent communication. So this is a real study that currently we are working on, and this is being produced and given to the sponsor every month. This has two numbers, and I just took off those numbers because for a lot of reasons. So here we are projected what is be the sites will be activated and what is the actual recruitment. And there is another graph that will say the projected recruitment too. That will be the projected recruitment, and this is the actual recruitment over there. As you can see, the bar graph. This simple graph that was produced using SAS is extremely important for sponsor to monitor this one. I am designing a trial. I'm a PI initiated trial, and I have a sponsor, and I'm telling him, oh, I can recruit 100 patients per month. He will say, sure, okay, I need $100 million. Perfect. Given the credibility of what PHRI has it and what Dr. Yusuf has done in the past, they will give you. But how do they monitor this one? This simple graph monitors whether we are sticking on to our commitment. This commitment is extremely important for sponsor and give them the confidence that we can actually run the trial. As you all know, randomized trial is truly a gamble. How much you study, how much you designed and everything, you cannot predict the outcome. You need to wait for it. You need to just be patient enough. But what you have control over is the monitoring of it to make sure there is nothing wrong happening in the data, not affecting the patient. So when you're doing it, and as a PI, you might not request for these kind of graphs. You might say, oh, sure, you have recruited 8 patients, fine. You might move on. No, you should request a statistician or study coordinators to produce these kind of graphs to show you whether the study team and everybody around the site are doing their job. So that's a part of the researcher to be proactively asking for this one. What's that? The sites activated number should be somewhere, not in this graph. Probably on the right side, yes. And there was also a legend there, but it's missing. So yes, on the right-hand side of it. And one of the first report that we create when the study is first patient is randomized is this recruitment by country. And this report is a weekly report, and it is sent to the sites and also to explain to them what they're doing. And when it's sent to them, it will be sent only their information, not the whole study information, and unless it's really required for them. So it might looks like a normal eight column report, but it would take at least a month for us to make sure the report is producing the right numbers. And especially the third column from the left is the average monthly recruitment per site. Someone were talking about the pilot studies. The pilot studies, what is it you are currently working on is the average recruitment. That is your primary focus. You never focus on your hypothesis. You always focus on, am I able to recruit patients? That tells you, to my commitment, am I able to produce at least 2 or 3 patients per week? And the sponsors are very strict about this one. You need to have a proper projection on how much you can recruit. And if you're not able to do it, they have all the rights to stop the funding. So this kind of reports, this is again produced in SAS, and as a statistician, it should be pretty straightforward to pull this one. And the softwares that we are using for the trial master, as Ananta was saying, we are using DF Discover, we are using Trial Master, we're using DataFacts. So sorry, DF Discover, Trial Master, and RedCap are the 3 different systems that we are having, the EDC systems. RedCap is very minimally used, and it doesn't have a whole lot of features that is required for a large randomized trial. This is a 8,000 patients randomized trial. And we have around 25,000 patient randomized trial. The RedCap doesn't have the capacity or ability to handle that. Its only reason is not because they cannot handle the number of records. It cannot handle the edit checks. The monitoring that we want to do is in different stages using the edit checks, using different type of reports. And especially the REDCap doesn't have that strong ability to have larger edit checks on it. So that's the reason why we are using the trial master, and sponsors and everyone require that. Any questions on recruitment report before we move on to the data quality? Feel free to ask any questions and stop me at any time. Disclaimer, I tend to talk fast, so please stop me if you think I'm talking fast. Yes.
So if I am working on clinical trial budget,
Yep.
resources
Mhm.
at my disposal
Mhm.
to do a fancy electronic data capture system,
Mhm.
what could I do?
You can still collect all the information at the old school at a paper copy with an Excel. And this is not going to stop you from creating a such report, Shrikant. The purpose of this is you can still go ahead and create all these reports, but it is little tricky. I mean, SAS is your primary software, then you can have it from Excel to SAS, and SAS to any reports can be done. The system is always preferred as a,
I think, no, I just wanted to clarify to
Yeah.
the participants, because we are presenting sort of the state of the art
oh yeah.
on how
Mhm.
we do things.
Mhm.
And not everybody is able to do that. But the recommendation is that whatever system you have that is available, you need to be able to produce reports for monitoring, but also you want to ensure that you keep copies of the original source data. Because if ever some regulatory agency decides you may need to be audited because they think that you may have committed fraud, you have to be able to defend yourself and have the original source information. So that is important. And these fancy, costly software does it for you. But if you don't have the opportunity for that, you still have to do it. You still have to follow good clinical practice, which means to have that ability to defend yourself and stand up to an audit. And you can do it with
Yep.
paper forms,
Yep.
you can do it, but it is time consuming. If you have the money, do it with software. If you don't, do it another way.
But this one is not from the EDC system. This is from the SAS software. We export the data and we programmed it to have it happen. So these are all more of monitoring aspect of it to make sure a successful trial. Yes. And before you want. Yeah, I'll show you a separate a DSMB report when I go to that topic. It's a live report that we are actually working on. Yesterday I sent a DSMB report. I'll show you that. This one is more of an internal monitoring, a study team who looks at it, but at a different level, we look at the different reports too. Yes. Yes.
Sir, can you just briefly explain the difference between ADC and FileMaster?
Yeah.
And I mean, in fact,
Yep.
even REDCap and
Yep.
SAS, okay,
Okay.
because I am
Mhm.
not familiar with
Sure.
any of these.
So EDC is the electronic data capture systems. EDC. So it's like you can collect in Excel, you can collect in DF Discover, which is data facts. You can collect in REDCap, you can collect in Trial Master, you can collect in Brave. So there is a whole lot of systems. And recently also, I think one of our colleague brother is also having a company, and he he has a company that provides EDC systems. They build their own EDC system. So there is a whole lot of things in the market where you can actually use it for clinical trial, which is cheaper and whichever is cost effective for use case. And softwares for analysis is what the SAS is, SPSS, R, Stata. What else? I think that's all, I believe. Is that? Oh, Python. Yeah, we can use that too. If you're, I'm a simple biostatistician, and learning SAS itself is a lot for me, and Python is too far away from me. So yeah, you can do that, definitely. But SAS has a whole lot of options to make it more prettier. These are all not manually entered. It's from the SAS data set to the report, actually. So we have different macros that can be used. So this data quality is extremely important, and there are several ways to monitor the data quality. And as Shrikant was mentioning, if you're not collecting the right data, and there may be an audit who will be asking whether you have collected the data properly, have you been audited, have you been properly keeping it, the records and everything. So we need to make sure that the quality of the data that you're collecting is being properly managed, and especially if there is 1,000 CRF collected and there is 10,000 points, is everything is being entered. There will be queries happening. That's why we prefer the other two systems than the RedCap, because in the RedCap, you cannot put a lot of queries on it. Like, for example, in a site enters, as someone Ananta was showing, that you entered 1,000 or 10,000 for a BMI by mistake, you need to put a query inside the system, and so that the other person can see it in live. It's like a OneDrive, or in a live, you can actually see what is a query and they solve the queries there. So similarly, we need to make sure that all the queries are resolved too. And how do you monitor all the queries have been resolved, all the CR has been collected, is using the data quality report that we have created. And as I told you, it took at least 20 years for us to come to the point. The report that I'm going to show you, it's one of a kind. It's a very detailed report, but it took us 20 years. It didn't happen overnight. There was a simple report that we were handling it, but based on the different audits that we have been receiving recently and the feedback, we have ended up in this fashion. And again, you don't need to have the same type of report, but you should be focused to have a report something closer to that one. And the importance is, again, the foundation of the integrity and the participant safety is the most important one. So there are different ways to data validation and the cleaning aspects of it, but the regular internal audits is extremely important. A lot of times we tend to not have an internal audits can be anyone. It can be Dr. Tambur, like it can be anybody who can actually do an audit for you, who knows about it, who knows about what FDA is going to look for it, what our audit team is going to look for it. So internal audits are very important. And recently we have been having 3 internal audits even in our team. So those are all going to help you. So this report is the data quality report by country, and it first column shows the randomized and the number of forms entered. As you can see here, there is 169,000 forms has actually been entered at this point, a number of forms without incomplete stages, and you can see each one goes there. And it is a live document. It is being constantly updated. The way the data comes in, there is a process of cleaning the data and coming here and then validating it. It goes in a circle, and it is a constantly done for, let's say, 8 years, your trial. All 8 years, you will be doing this every single day. It is not, and that is, if you do that, the quality of the trial that comes out will be amazing, and no one will question your credibility or the trial that you're being publishing. Any questions on this one? Yes. Everything is automatic. So there is no manual. We can download anything we want. It's a system, and we convert that into a SAS. I can actually download every single query and look at it too. But we have a system that we can actually look at it, and it is automatically transferred into a SAS format, and we produce this report. Yeah. 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 Yeah.
Because that's the problem. What we are facing right now with development of our Shiny monitoring.
Yes.
Reports.
It is tough. That's
a few places you can automatically download things using API, but at few stages you have to go and click. Yep. Because RedCap, because it's a low budget software, but it's not actually the problem with Cleaner and Zelta.
Yes, it is tough. It is tough, especially with RedCap, and you cannot do a whole lot of things. And that's, again, the reason when you're running a large trial, people try to stay away from RedCap for all of these reasons. But definitely the other systems allow you, even sometimes you can even create a simple report within a system itself. Even you don't need to download into a SAS and create it. It comes out a system, but it will not be as comprehensive as you can see here. Because as you can see that one column says number of queries and bypass nodes assigned to PHRI role. You will not see that in any other studies, or it's very unique to PHRI because the sponsor who have given us more than 100 million, or I don't know how much is it. They want it like, okay, there is a query has been assigned. Are you doing a job? So it's sponsor who is monitoring this report. And if that one is high, you're going to question it. Oh, you guys are not, you had 8 FTE to do a trial, but what you're currently doing is less. Yes. Yeah. Okay. So in a system, so as Ananta was mentioning, if you have a BMI, you need to enter the data. Okay. You need to enter between 25, 30, or whatever it is. You need to manually enter the data, or you need to scan the document. It will automatically populate in the field. So if you have seen a EDC, have you seen a EDC system? No. So you have seen an Excel, right? So imagine you collect in an Excel and it will populate, and your age shows up as 185. It could be a or let's take a weight. It says 305 or something. How is it even possible? Maybe they are entering it is when they enter 30 or something, they entered it by wrong. Now we have different reports. I'll show you later. There are different reports that we will be looking at the quartile ranges and min, max, and everything, and we monitor the data. And at the time when we notice there is an extreme value like this, we go to the system, because imagine a site is in Africa who's enrolling patients for my trial that I'm conducting in India. How will I communicate? I communicate through the system itself. The system has an option where it will go to that field, you right click and you tell them the field is outlier. Can you please check? What the people in Africa do is they get a notification. They take the source document, the actual source document that Srikanth was mentioning. They look at it. Oh, it's 85. We made it wrong. So they go into this query, they enter it, they solve it, and that's when the query is solved. So each time that happens, right? Like, and you can do it in a small level at RedCap too. It not required this whole fancy stuff. You can do it at a small level, but you need to monitor everything. And it all done centrally. You're not going to sit with somebody and ask them to do. Okay. So I think the internal audit for us, we do it every 6 months. It's a lot of work again to do it. And especially it's a sponsored trial, we do an internal audit for sure. But if it is a PI initiated trials, and if it is not been IRB approved, or it is not going to be submitted for any drug or anything, we do it, depends on the requirement. So that's another thing that Srikanth is currently working on. As I said, clinical trial will challenge you every single day. Now Srikanth is working on designing our whole SOP, and the topic that the people were talking about, the standard operating procedure. We are going to divide that for sponsored trials and non-sponsored trial, which is internal trial, because you will know more in an upcoming slides why we need to have a different standard operating procedures. Okay, and then compliance with the protocol, and the importance is, as you know, the protocol dictates and it tells everything. As I forgot, I don't know his name, but he was asking about what if there is an inclusion exclusion criteria. If you're not following the protocol says you need to have such an inclusion exclusion criteria, you need to follow that. You need to have a history of hypertension. You should not have something, you should have something. Everything need to follow the protocol. So if there is no protocol has been followed, then it becomes an even more complicated to run the trial. And that is the most important thing. They will question it. And in an audit, you will be in a difficult spot. So that's, again, the regular monitoring and the audit process and the compliance identification will tell you how this need to be done. For example, let me give you an example, especially the protocol one. When you are randomizing and you have a randomization list, PHRI is very unique, because we are the only institute where the DSMB, which is Data Safety Monitoring Board, and the study trial that we conduct is happening in the same building. In generally anywhere else, you will be the host, you will be running the trial, but you will be giving it to a different institute, a CRO or a pharma or any other place to run as a DSMB, because they want to keep a firewall to make sure no one disturbs them and no one knows anything. But Dr. Yusuf felt, no, it's not efficient. It's also expensive at the time. So why don't we build a proper firewall within our systems? So when we do this protocol adherence, one of the things about randomization, you need to monitor whether the randomization is properly allocated. Someone was asking about the 2 block and 2, 4, 6 block size, and you need to make sure the allocation is properly done. Those things are monitored by an unblinded statistician, where we will not be telling others, but we say a system is properly allocating people. Imagine at the end of the trial, we have 100 patients, 10 of them in one arm, 90 in other arm. You're screwed, you are gone. And who is going to monitor it properly and constantly every week? The unblinded statistician does it. That's part of the protocol adherence. If you're not following the adherence to the protocol, then you will see there is a deviation happening, which will come up in a second. This is randomization irregularities. This is another report that we produce. This is constantly produced for a DSMB, not everyday process, but this is produced to a DSMB to explain, okay, we have tried to click on a system, but we randomize the same patient twice. Okay, then what happens? Why do we need this? Because it tells you, you randomize 2 patients, what happens? It takes the next allocation for that patient too. So you are wasting an allocation. Now there is a deviation happens. Vignesh gets the first allocation, he's getting a treatment. And the next allocation in that is, let's say, 4th block, and I'm getting a placebo. I'm supposed to get a placebo, but he was randomized twice, then I'm wasting an allocation here. So you need to be very careful about what you're doing, and you need to record that one. The audit will ask you how many deviations have happened. What are the different type of irregularities? The site sometimes clicked twice wrongly. And someone was talking about stratification. A lot of times when you click on a system, when you're doing it, you click on a wrong stratification. Every single wrong click on a stratification gives you a double randomization wrong. Like it just creates a problem. So everything need to be recorded, and the protocol deviation too. For example, as you were asking, is about the inclusion and exclusion criteria. And it says here, there are some numbers that say not completed per protocol. And you have number there is not 0 there. And now we have a line listing after this. It will be produced and given to the DSMB to monitor, to make sure that that is not a problem. If there is a problem, then they will immediately tell the team that you need to be careful about this. And if it is going to continue, then there may be a chance that trial will be coming to stop. So everything need to be recorded, because this is an FDA submission trial. The one which I'm showing you is going to be submitted to FDA. So that means the process involved is extremely high, and the quality need to be high too. Yes.
So whether DSMB is applied only for multi-subject trials?
I'm going to come to that slide soon, but no, it's DSMB is there for any clinical trial. I'll go over the history of DSMB, and in that you will see it's applicable for any clinical trial at this point. So adherence to intervention is extremely important, and I will go through this one here. So adherence to intervention can be mainly giving a proper training to the site or the people who are giving the drugs. Because if they don't know what kind of drug and where to take the drug, sometimes there will be an injection, there will be a tablet. So you need to be extremely careful and properly trained when it comes to this one. This study that you're looking at is an open label trial. So Shrikant, have you spoken about the open label yet? No. Okay. Let me not confuse you all, but it's a phase 3 randomized trial. As you can see, double blind, and double blind is what Ananta was talking about. But this one is a single blind, and it's an open label, what we call it as, because it's a surgery trial. The surgeon who is doing it knows what he is doing. He cannot be hidden. It's a LLA occlusion. It's a device that you need to put in your heart, and you need to do that. So he knows exactly what he's doing. Is he going to do the LLA occlusion or not? Because you get randomized to that, and the surgeon knows it. But the site, the coordinating center is completely blinded, and they don't know any of these informations. As you can see here, this crossover, the received intervention as assigned, and you can see that the number might be very close to the people who have randomized on that group. This is the overall table, but we will produce a similar type of table with overall by treatment too. And then you can see a crossover, and the crossover will have a line listing. And then the last 3 columns, 3 rows that you're noticing, it is all 0 there, but I just put a black mark there. But these ones are unblinded information. Here it says left atrial appendage exclusion attempted, What does that really means is you will know who is being given in what treatment. So this information is not disclosed to anyone. It's an overall report. And you might see a topic there called intent to treat. That is covered, Shikan? No, not yet. Okay. So he'll talk more about that, what is called ITT here. There is an ITT, there is a per protocol, and then there is something called as treated. Perfect. What's that? No, so it's not after the surgery. So what happens is when the people have been trying to attempt the surgery on, let's say, LLA occlusion, there may be different things happens in human body, maybe more bleeding, something happens, and the surgeon decide, no, I'm not going to do an LLA occlusion for this patient. I'm going to just do it the other one because it's not satisfying the condition. Even though the inclusion criteria, everything satisfies, but when you do the surgery, there may be a change. Or patients in the surgery bed, like before going to the surgery bed, they will say, no, I'm not doing it, because patient will know what surgery they are going to get it. They will say, no, I'm not doing this one because I don't want you to do the device. Now, the patient is going to be still in ITT. That Srikanth will explain what the ITT here means, because they are randomized. But now they have crossover because you're already in the bed. Now I cannot do the LLA occlusion. So I need to do the normal procedure, which is very important for the patient. So they will do the normal procedure, and that's where the crossover happens. Unblind crossover, yeah. So next is the safety monitoring, and safety monitoring is related to the DSMB and especially the adverse event and the safety. So when you are doing an FDA trial or a trial, it's a large scale, there is a requirement to report the adverse events. In a lot of cases, adverse events are mortality. It happens, but in other cases, there is a lot of other adverse events that can happen, especially with the surgery. Bleeding can be an adverse events. With a hip surgery, bleeding is another, and stroke can be in one of the stroke trial, the ischemic stroke can be an adverse event. So a different type of adverse event happens, and they want to constantly monitor. So that reporting of adverse events is very important, because you need to know what is an adverse events. And as soon as you see that in the database, it needs to be reported to the concerned committee. And we will talk about the committee here. We will be talking about the steering committee. We will be talking about ethics board and other things, and they are supposed to receive a copy of every single adverse events or the serious adverse events that happens. So as a coordinating center or the coordinators, you need to be careful about what it is. And this is a very important thing. When you're running a trial, they will ask, okay, have you observed the adverse events? That's the first question they will ask. So this is, I was mentioning about the MEDRA coding. As you see there, it's a MEDRA, it's a system organ class. And this is based on the MEDRA coding. And I was reading what is the difference between the ICD and the MEDRA coding in one of my phone or my laptop, but I'll share that information. But this is a more generic one where internationally there's been used to know, like blood loss anemia. All of these need to be categorized into a particular category, and that's called lower term level. And this is the one which we are using, and this is being reported to the DSMB members, and also a line listing is given to any board that is audited everything. Any questions on this one? Okay, so the last one for the internal monitoring, and this we did probably, Shrikant, to 2017. I think the year you joined PHRI. So my previous director, and she's no longer, and she died in 2016. Her name is Dr. Janice Pogue. Her PhD thesis was on central statistical monitoring. I will share the paper that she has developed and the risk score. The most important thing as a statistician is to run a simple check on how to monitor the data in a statistical way, in a proper statistical test. We are doing a multi-center sites. So what happens is we are having a common inclusion criteria, common exclusion criteria. All the information that we are adding and the randomization is to create or remove all the bias that we can have. So that means all the sites around the world should behave almost the same, because that's what the clinical trial and the randomized trial, the beauty of this one. If they're not behaving, then there must be a reason behind it. So we divided that into, I have a separate poster for you, but just I want to quickly run over this. we have a different ways to find it. Is it too good to be true? So we divided that into a good, bad, and ugly. So good could be, we think it's bad and we look at it and we say, no, everything is fine. For example, we had a site where recruiting patients on a Sunday, like 10 or 9 patients on a Sunday. Every Sunday, you will see 8 or 9 patients being recruited. That's weird. How is it possible that a particular site, when we compare it, the site data with the rest of the site, they are on time exactly on a due date on a follow-up, and the recruit on a Sunday without something is going on. Okay, when we asked them, they said due to limited resources and everything, they arrange a bus. So when they identify the patients, they go to their home, pick them up, go to a particular center, they randomize, get all the data and everything, and they enter the data. It's all on Sunday. So they're not doing any fraud here. But are we supposed to monitor? Yes, we are supposed to monitor that. Why? Because one thing happened with the bag is we had a SBP, the blood pressure that you measure. We had a variance checking between a particular site. They are completely different from the rest of the site. Okay, now we started looking at the site and saw that blood pressure is between 120 and 130. Dot on between 130 and 130, we have around 40 to 50 patients. How is it even possible? No, it's not possible. So now we asked for the source document. They showed us a source document. It says they measure a blood pressure for a patient at least 10 to 15 times until it comes within the range of 120 to 140. It's a wrong understanding of the protocol. It's not that they're doing a fraud. They thought we put a range called 120 to 140 for a patient. They thought all the patients should be between 120 and 140, and they keep measuring that. They make them to sit for hours and hours together to come within the range. How we found it? It's just simple statistical technique. It's a simple chi-square test. The t-test is going to just simply tell me this site is completely different than the rest of the site. And that triggered that one. Ugly is a proper fraud that we can find. When I say fraud, it's truly a fraud, and that's why I'm using that word. There is no patients exist. When we found out the site is completely different, it's too good to be true. When we looked at the source document, that's why Shrikant said you need to save the source document for a lot of reasons. When we actually looked at it, there was no patients. Now, what happens if we didn't monitor this one is we need to remove these patients. They are not going to be, just because they are randomized, that doesn't mean we need to keep in the study. They are going to be removed from the study. Now go back to the sample size calculation. You have already said you need 180 patients for your trial to have 80% power. I am removing 40 patients from this trial because they are fraud. Now I have 140 patients. It's an underpowered trial. Everything is waste. So now, if you have found this early, you can recruit more people. So this monitoring is extremely important, and best use of the statistical simple techniques. It's not very clear here. It's a basic randomization baseline, and it's a simple chi-square t-test, and F-test is what is being used. It's not a very fancy risk score as my director was created one. So it's extremely important. Yes. So what we do is, let's say it's a site 1 and compared with the rest of the site. So you have 50 patients in this, 50 patients compared to rest of the sites, and you say, is it statistically different or not? It's a simple score of 1 or 0, because if you're significant or not. And then we can also do a fancy one by giving a weighted means and things like that, but mostly it is a pretty straightforward one. Any questions on this one before we go to the next topic of DSMB? Good. Okay. So yeah, conclusion, internal monitoring is extremely important. You need to use the best resources that we have, statisticians or programmers, make them to work on different type of reports, and they will be happy to do it. But you need to think through the process, and the most critical person or the group during this internal process is a study team. Is a team that is coordinating the whole study, is the most important individuals in this one. Okay. So external monitoring is extremely critical, and it's definitely a privilege, especially because you are unblinded to the treatment, and there is only 4 people or 5 people in a DSMB, not 4, it'll be an odd number. So it'll be 4 or 5 people in a DSMB and one unblinded statistician. So there will be 5 individuals who are privileged to know the by treatment results. They are the ones who are going to recommend to the whole group of people, 100 to 200 people who are working on a trial, to tell you can continue the trial or you should stop the trial for safety, for efficacy, for futility. I hope everyone understands these terminologies, but if I can explain, efficacy is you are looking at the superiority, you're just making sure that, okay, one treatment is working better than the other, you stop the trial. Safety, there is something bad happening, extreme bleeding is happening in the trial, which is expected, but still is extreme numbers, a Z values like crazy with zeros, stop the trial. Futility is you're not able to recruit people. The recruitment is extremely challenging. And is it right to put the patient through the risk because you don't have the ability to recruit? Then the DSMB can recommend also to stop the trial for futility. And there is a different criteria, and Shrikant will be talking more about Thursday on when to stop, how to stop, and that's going to be a very interesting topic. So here we are going to cover primarily about what is the steering committee, and I'll go through a lot about the DSMB, the process, how wisely you need to choose the DSMB members, and then what is sponsored and ethic boards. That's a final topic. So steering committee is an expert composition, and they are blinded. So they don't know by treatment results, but they are the experts from around the world. So they are biostatistician, there will be ethics, and everybody will be there in that committee. That's the composition, and every trial should have a steering committee group. And they have a very comprehensive oversight, and they work very closely on approving the protocols and a whole lot of decisions. So the first function or the oversight responsibility they have is approving the study protocol. The steering committee plays a critical role. So the study team, the PI writes a protocol with thinking through all the problem, and then it goes to a steering committee, they present it, and then they are the ones who need to approve whether it's going or not. And then the monitoring of trial progress. So there'll be a constant steering committee meeting probably every 3 months to 6 months. You need to present a whole lot of comprehensive reports and telling them the trial is being recruited as per the protocol. What are all the deviations that is happening? Am I doing the right thing? Is there anything new that is happening in the market too? So there is a different terminologies, and just not to confuse you, so I've just written it down. So people call it as DSMB. Sometimes they call it as IDMC, and they call it as DMC and DRB. These are very interchangeable. You can use it at any way that you want it. In US, a lot of times they call it as DMC for some reason, and they call DSMB. You can call it anything you want, but end of the day, they are going to monitor the data that for efficacy in an interim and mainly the safety at a constant interval. So to answer your question about the DSMB, and in 2006, there is a guidance document by FDA, and still that is being used as a Bible for DSMB. And this is a group of individuals who scientific expert on that one. So they have defined it, and that is the document that is being followed by all the institution, like that includes research institute, CRO, and also pharma. They review on a regular basis, and how regular it is, we need to figure out, depends on the institution, depends on the PI, we put it in the charter. It can be every 6 months, sometimes it can be every 3 months, or it can be very ad hoc in the way that the DSMB members can ask for every week report too. I will go through that in a little while. So advises the sponsor and the researcher regarding the continuity and safety of the trial. So DSMB is very crucial, and it is ethically right to have a DSMB. Do not run a trial without a DSMB or a monitoring committee. Sometimes we also have what you call as a data monitor. There'll be only one individual who will be just looking at your data because you don't have money or you don't have the capacity to have people around the world to join that one. So what they called as a data monitor, who will be an individual from the same institution who has a knowledge about this one, who can actually look at the by treatment results and tell you, continue the trial. They will not disclose any information, and they're not supposed to disclose any information to it. So the ICH E9 on the statistical principle, the independence of the IDMC is intended to control data sharing of important comparative information. So comparative is the by treatment results, and to protect the integrity of the clinical trial from adverse impact resulting from access to trial information. So in a lot of cases, when you have a sponsor, it's not to criticize the sponsors, but they have invested a lot of money. They want a very successful trial. They want the drug to be in the market. So they will try their best to get more information or close the trial early with benefit. Is it ethically right? No, but they are also investing, and they will try anything and everything as possible. And that is a challenge always there when a research institute is working with a pharma. That's why pharma tries to work closely with CROs, because they design their own trial, they give it to CRO, and CRO does anything and everything what the pharma needed without questions asked. But research institute, we fight back. We don't tell, no, I'm not giving you any information that you're asking for. Don't ask me for any information. I don't even talk to them. I'm the manager of the Department of Statistics, and I need to handle 40 projects, but I keep the firewall so well that they won't even be talking to me for any reason, operational reasons too. Because I am the DSMB reporting statistician, then I won't be talking to them. So we are very strict on that aspects, and which is very important. And that's why the research institute are ethically always right, to my knowledge, or with my experience. You take Duke, you take NIH, you take North Carolina, or you take PHRI, any research institute, they are really good on that aspect. So quickly, I'll run through the slide. And again, we are in a very prestigious institution and CMC Vellore, because they have been a pioneer for biostatistics on a clinical trial in India. And likewise, North Carolina, I was just telling to Shrikant, whenever I do a research on something about clinical trial, first thing that comes out is the University of North Carolina. They have been a pioneer for clinical trial, and especially to do with biostatistics, their names comes out. And as you can see here, Dr. Bernard is the pioneer who have developed a document, especially for to have a DSMB, a proper DSMB, and formally published in 1988. And as you know, the first sponsoring for multi-center trial in 1960 is also by NIH. And during even recently in SCT, in Society of Clinical Trial meeting that me and Shrikant were in, they were talking more about DSMB, the importance of DSMB, and how important is to have the independence for the members to do their job. Yes, the DSM members are well trained, because a lot of people have been like highly experienced. Now the next generation has not been trained enough to be the members of it. And I was even asking Shrikant, so I should be part of a DSMB committee, because that's the next step for statisticians. It's important that you can actually volunteer to be a DSMB member, sitting and observer for a trial, and you will learn a whole lot of things. That is a need of the hour. And it's a very important agenda in SCT is to create a committee to train next generation statisticians to be a DSMB members. A good DSMB members can change the course of the trial, or a bad one can spoil the whole trial, because they are the ones who are recommending to stop the trial or continue the trial. Recommending, the word is recommending. They are not telling you to stop. They will just recommend you. But to defend, to say, oh, no, I'm going to continue the trial, you cannot do that. Still, you need to have a justification why you want to continue the trial. So DSMB is extremely important. And that was felt, 1960 itself, like they felt is very important. And as you can see here, protecting the participant safety, ensuring the credibility and integrity of the trial for the future subjects, ensuring the timely conclusion of the trial, identify protocol violations, identify unexpectedly high dropout rates that threaten the trial ability to produce credible results. And that's an extremely important one too. And they observe all those things during the DSMB report and every 6 months when they're looking at it. And they ensure the validity of the study results too. And that's why in every DSMB, we will have a biostatistician in it. The reason of it, and a lot of DSMB, the biostatistician will be the chair of the DSMB too. because they can look at it in a different perspective. The perspective is not just the data, it's about when and what and how they need to be done. Shrikant is part of a lot of DSMB as a chair too. So usually the DSMB members are created and they are being initiated by the sponsors or the people who are initiating the trial. So choosing the DSMB members is a critical aspect of it. Dr. Yusuf doesn't like, when I say Dr. Yusuf, just for information, Dr. Yusuf is our former executive director of PHRI and he's a founder of PHRI. So he doesn't like when the trial stops early. He wants trials to continue. And he is very cautious and very respectful how you choose the DSMB members. They are volunteering to be part of the research institute. They are not being paid even a penny to be a DSMB member. Sponsors, when you are a DSMB member for a study that is sponsored study, sponsor will pay something. But all of the individuals around the world, they are volunteering. Why are they volunteering? Is for the patients, is for the betterment for the trials. Their time, at least every 6 months, they need to spend 2 hours and reviewing the report, writing the reports, and then making sure they give a proper feedback. So choosing a proper DSM members are extremely important. And it is a very, we need to be extremely cautious, and you will see how detailed our DSMB reports are because they are spending the time and effort for the patients, and we need to be very proper as a statistician to make sure we give them every single detail and not to hide it from this. The function of the DSMBs, as I said, they need to make sure that the formal written advice to the sponsor and the potential protocol modification needed prior to the study initiation. So DSM members can provide their feedback before the first patient is randomized. Once the first patient is randomized, they are unblinded. They cannot give any feedback about your protocol, what to change, what not to change. Recently, there was a trial we had where we had an outcome where DSMB has looked at it. Unexpectedly, the efficacy is in our favor on the treatment side, but the PI, who is not blinded, who doesn't know anything about the trial, thinks, no, something wrong is going on. I need to change my outcome. Now, who can say it? I'm unblinded. I know it. My DSM members knows it. But we were just sitting there. We are listening to him. He just wants to change the whole outcome. We are doing a first primary outcome. He wants to do all fancy things because he feels it's underpowered study. There is nothing significant happening. There is no use of it. I'm going to change that. But we cannot say anything. That's the beauty of this one. They will not even say a single word to give you a hint that, oh, don't change it. Your trial is going to be positive. You cannot say that. It's ethically wrong to say it. But is it affecting the study? That's why the researchers are highly respected, is because of the thought process. They need to think through properly. They should not be taking decisions in ad hoc, right? So, but the good DSM members won't relieve any information. They won't tell you. They won't, because they don't work for you. They work for the patients. They work for their health, right? So, yes, yeah.
If the protocol is not yet finalized,
Yeah.
and there is, can a DSMB member ask for the protocol from the
So what happens? I'll show you the order of the DSMB meetings, but the first meeting happens where they will be discussing about the study. Once they find the DSMB members, there will be a meeting that is happening where they will be presenting about the study, sharing the protocol, sharing the charter, and they will be sharing the mock shell tables. All of these are shared with the DSMB members for their feedback. They can provide any feedback they want because they will be asking what is happening around the world, what is happening on a cardiovascular, what is happening on a coronary revascularization on that trial, because they are the ones who are world leaders on different aspects of it. They know what is happening in different places. So they will say, okay, currently we have a colchicine study that has said there is no benefit in this treatment. Why are we testing the colchicine here? Are we sure we are? Then they will say, no, this is a different type of population. So that input can be given. That only before the first patient is randomized. Once it's randomized, you are only a listener on this one, on the other side. What's that? Oh, yeah. Yeah, because the protocol won't be finalized. But once the protocol is finalized with the ethics, and that's when we first have our DSMB2, because we cannot randomize a patient before the ethics has been cleared on the document. So once the document is cleared, then that's when the first patient is randomized. But during that, between the time is when the first meeting happens. Some trials are even more stricter that they need to have a charter and also the SAP for the interim analysis too. The sponsored trials, you need to have a SAP before the first patient is randomized. Yeah. Yeah, so let's take ours as an example. So PHRI, we have a lot of sponsored trials, and we are the own sponsors too. Like we are the PI initiated trials, and we select the DSMB members. As I told you, Dr. Yusuf recommends, he knows around the world who's are all the best people around it on this particular disease, and we recommend and we talk to them and say, can you be part of it? But they don't have any benefits of just being part of this study, and it's only for the betterment for the study. Yeah. Yeah.
Has it to be endorsed by some other agency?
No, technically no. You don't need approval from any other institution that you are forming a group of 5 people that you know of. It's fine. You can still find anybody whom you think who is going to be ethically right, who's going to help for the study. You can select them. Yeah.
Who's part of DSMB, he might be.
That challenge is always there. And even when Shrikant joined at PHRI, he was even surprised the fact that we are holding the DSMB itself, first of all, at the institute who are running the trial. But it is that credibility that you need to build. Initially, people won't question it, but if you can maintain that credibility, to prove the world that I can actually do it by having the same people in the same room, I will not discuss anything. That takes time to build it. But yeah,
the different sponsors will act differently, right? So pharma wants to, when they're sponsoring a study, they want to show that the DSMB members don't have conflicts of interest. They're not going to be burdened by the fact that we are asking them to do it. They're independent and so forth. So they usually are very careful in who they invite. They also have legal documents where the members will sign that they have no conflict of interest and so forth. In non-sponsored trials, non-regulatory trials, there is a little bit more leniency. But as Kumar says, you want it to be seen, or maybe the optics of it need to be clear, that these people are working independently. Yes, I may have chosen them because I know them, but we also have them sign a document, it's not a legal document, that they don't have any conflict of interest in participating in it. And usually it is an honor to be invited to be a member of a DSMB. So most people that are asked to do it actually do it because it's an honor to do it. Or as Kumar says, for the betterment of patients, et cetera, altruistically, they do it. It's a great thing to have on your CV, guaranteed. It's a great thing because also, as you will see when he's presenting, that the discussions that take place during a DSMB meeting, especially in the closed session, are extremely enlightening, informative. It's a learning process because the members of the board are statisticians, clinicians, epidemiologists, behavioral scientists, others, multidisciplinary. They come with different viewpoints. They're looking at the same study. They're looking at the same efficacy, safety, futility, and they come from different viewpoints. The discussions, I learned a lot when I was in the first DSMB. And now other people in the joint, just by sitting down and learning or hearing how the discussions go by, it's extremely, extremely enlightening. And so members hope to be invited to join. And usually people are not paid. Pharma tends to pay. And there is this issue we were talking before of paying participants is maybe a coercion and goes against informed consent if you're paying them a lot to participate in a trial. Pharma pays people to come to be on the board, and they pay a lot. And so now you think of a conflict of interest by the payment, because if we stop the study early, we don't have any more meetings, we don't get paid a lot again, so I'm not going to stop the study early. So that's a big issue. So the governments don't pay. The NIH, they pay an honorarium that's a minimal, the $100 a day or something like that. But in the US, $100 is not enough for anything. It's one dinner someplace, and they pay your travel. Nowadays, they don't pay anything because they'll just give you $100 because everything is over Zoom. And they found out that it's cheaper to do this over Zoom than to have people travel to Washington. But pharma still continues to pay. And so people that join DSMB boards for pharma are, we worry about conflicts of interest in that. They're not getting, I can tell you an example. I should not tell you an example.
Yeah, just be careful. Yeah. So that's where it will head to. Like, it's very difficult, right? Like what to say, what not to say, and especially when you're doing it, and it comes with the practice, I would say. Yeah, I mean, even now, Shrikant sits in several DSMOs. I just sent a DSMB report for a member. You ask me anything and everything, I will not say even a single word about the trial. I will try my best not to even remember about this one, so that by mistake also I won't say anything. So the most important aspect of this, before the first patient randomized, is finalizing the DSMB charter. The charter is the one which is created or developed and approved by the sponsor or the institute who are running the trial. And it will be sent to the DSMB members, and they need to approve it, because there is a whole lot of factors that will be there. That includes a conflict of interest and their roles and responsibilities, structure of the meetings, and guidelines for publications, because a lot of times the DSM members don't get the authorship in the main publication. Some do, most of them, they don't, they are not supposed to. So they put a very disclaimer saying that you will not be part of the authorship. And then rules governing the confidentiality and communication. In a lot of meetings, as you will notice, if when you're part of it, is there are some DSM members who will say, please do not take the minutes. The reason why they say is in US, there is a whole lot of issues around suing a company for running a wrong trial or drug or things like that. It's very, very common. So the people from US, especially the physicians from there, they say, nope, we're not taking any closed session minutes, because that minutes will be kept against them or used against them during the trial. When I say trial, it's a law trial. It's a the trial. When they go, they want all the records and say which person said to stop the trial, who initiated, and there has been several cases on it. So there are people we are very cautious about, and they will be very careful about it. So the DSMB charter is extremely important. I will just show you this sample one. And what is their responsibility in the charter is, from top to bottom of the charter, the DSM members need to approve it. Because it has every single information about the frequency of the meeting, at what date you will be receiving the data, and when we take the snapshot. And I say every 6 months, or is it every 3 months we will have a DSMB, or when is the interim analysis going to happen? Recently, there is a DSMB member who asked us for guidance to stop the trial for interim analysis for the co-primary outcome, which has never been a case. And that was a challenge for us. And Shrikant and another statistician worked on it, and we published a paper on it now. And we have a special interim analysis rule for a co-primary outcome, and that is included in the charter itself. So the FDA-related charters, FDA-sponsored or FDA submitted trials is extremely a detailed charter, detailed SAP, and detailed protocol. But if it is a non-sponsored trial or non-regulated trial, we have a different template, which is very little simpler. You don't need to go in that detail of writing every single aspects of it. So the charter is going to be in a very important information, and it need to be properly followed. Whatever you write in the charter, it need to be followed. For example, if you say every 6 months you will have a DSMB meeting, you need to have a DSMB meeting every 6 months. You cannot miss it for whatever the reason. You might have a marriage, you might have anything. You need to be in that meeting. Meeting should have been organized and completed. And I don't know if anyone of you have been in a DSMB. To organize and complete a DSMB meeting, it takes a 2 months of an effort of around 8 to 10 people, 3 statistician, 5 or 6 study team, and then a PI. So around 10 people need to work for 2 months to complete one DSMB meeting. So it is a lot of work, and it is extremely important to you, because those are the meetings where the DSM members will say whether to continue the trial or not to continue the trial. And why we are doing this? To be ethically right. It is not FDA requirement to have DSMB for everything. Recently, remember there was a meeting, we had an audit meeting for a big trial. There was an FDA audit person was there. There was only 7 auditors around the world, sorry, around in US or in Canada. So he was mentioning, you guys have said every 6 months you have a DSMB, but you have not followed every 6 months. It was 8 months or 12 months and things like that. Why did you write it like that? You don't need to. FDA had given a leeway that you don't need to have it like that. You just write it to your own convenience. It's nothing wrong in having DSMB every 1 year. But don't give false information and don't follow this one. That was an observation. Luckily, he didn't put it in a rank 1 if it would have been bad, but he put it in a rank 3, saying as an observation. So you need to be very careful. As you can see here, it is not appropriate for DSMB to provide general scientific review or a protocol approval. That's after the randomization. After randomization, they should not be giving any kind of input on it. So the charter is, so for a non-regulated trials, we are the sponsors, we are the PI, so we prepare it. And if it is a sponsored trial, the sponsors are in charge of preparing the charter, reviewed by the PI and the team, and then goes to the DSME members for their approval and the signatures too. So charter is one place where the DSME members sign it. Yes. Yes.
Now for India, for that's it actually. I can't find anything in the ICMR national ethics guidelines. The details of the DSMB charter. So a certain template.
I just took out the charter. I think you were the one you were asking the question about the SAP template, or you were asking.
I was asking, yes,
You
I was
were
asking for generally for a
template,
trial,
right? Yeah.
and specifically, yes, the SAP template, and
Yes,
then
I just took it out from the NIH. NIH has published a template on all of these 3 topics, and I'll just show you now. We have a template, which is what we are using it right now for our sponsor trial. It's extremely detailed. That means all the sections need to be filled in. To fill in that information, you will take, to my knowledge, to write a SAP of that detail, it will take around 6 months to complete a SAP.
So if you could share it.
Yeah, no, I'll share it.
Thank you.
Sure. To answer the question, yeah, okay. So this is the orientation meeting that I was talking about, the first kickoff meeting that happens before the randomization happens. So primarily to
even they say they don't, they're not under your control, and they may tell the study investigator what's going on. So that's one of the problems. You don't want, so in general, you don't give information to the ethics boards. If they insist on it, that's an issue, right? And
Yeah.
then you have to put that in your manuscript. Transparency of who saw the data during the study.
Yeah.
One institution for he can be he or she can be a DSMB member of some other.
Oh, yeah,
Yeah,
it could be for another study someplace else, no problem.
And a lot of people want to be on a DSMB because they get an inside information too, because they are part of the DSMB. They know what is happening on the cardiovascular. They are specialized on cardiovascular. I'm on a trial. I know this big trial is happening. I'm a DSM member. I know all the data, but in parallel, he's also running a different trial too. Now he will be thinking, God, did I design it wrongly? Because I see the effect is not happening yet. But he cannot go and change it too, because he's just speculating that his trial is not working or it's working. Because he is also blinded on the other side of it. But at least he gets some more information on that aspect. But so in conclusion, this whole monitoring aspect of it is extremely important, as I would say. Yes, sorry.